Pharmapal.
From cross-docking to protein partners, identifying binding sites and predicting interactions. Hello everyone! Welcome back to our channel. Today, we're diving into an exciting research paper titled From Complete Cross-Docking to Partners Identification and Binding Sites Predictions. This study, authored by Chloe de Quecker and her colleagues from Sorbonne University, explores how proteins interact with each other, which is crucial for understanding biological processes and improving medicine. First, let's talk about protein interactions. Proteins are like the workers in a factory, each performing specific tasks. To do their jobs, they need to interact with other proteins. Understanding these interactions helps us grasp how cells function and can lead to breakthroughs in drug development and bioengineering. However, gathering clear data on these interactions can be tricky. The existing data is often heterogeneous, meaning it varies widely, and can sometimes be noisy or biased. To tackle this challenge, the authors propose a novel approach using molecular cross-docking. This method treats proteins as rigid bodies and simulates how they might bind together. By analyzing hundreds of proteins in their unbound forms, they aim to identify potential partners and predict binding sites. Key components of the study. Coarse-grained representation. This means simplifying the complex structures of proteins to focus on essential features. Scoring functions. These are mathematical tools used to evaluate how well two proteins might fit together. Interface quality. The researchers examined the size and quality of the binding interfaces, essentially, the surfaces where proteins connect. The results were promising. The study showed significant improvements over previous methods. The authors found that certain factors, like shape complementarity and physicochemical properties, play crucial roles in predicting how proteins interact. They also explored how proteins can have multiple binding modes, which means they can interact with different partners in various ways. This flexibility is vital for understanding the dynamic nature of protein interactions. Interestingly, the study compared their docking approach with a sequence-based deep learning method. While both methods have their strengths, the molecular docking approach provided more detailed insights into the 3D structures of protein interactions. Next, we'll break down the key figures and concepts discussed in the study, making it easier for you to understand the intricate world of protein interactions. The authors aim to enhance our understanding of protein interactions, which are crucial for biological functions. Table 1. Default parameter values. Purpose. This table outlines the global default parameter values used in the study. Key points. The researchers analyzed 72 possible parameter combinations. They focused on the top 15 combinations across the entire dataset and its 8 functional subsets. This systematic approach helps in understanding how different parameters influence the discriminative power of the docking method. Figure 3. Influence of parameters on discriminative power. Overview. This figure illustrates how varying parameters affect the identification of antibody-antigen pairs. Key observations. Distance threshold. The study found that adjusting the distance threshold between 4.5 and 6A didn't significantly impact discrimination for most functional classes. Preference for smaller DIs. Interestingly, smaller docked interfaces, DIs, were preferred for identifying specific interactions. This suggests that while some parameters have minimal impact, others are critical for accurate predictions. Scoring strategies. The researchers employed 4 main strategies to predict interfaces, each incorporating 3 to 4 scoring schemes. Here's a quick breakdown. Descriptors used. Evolutionary conservation. Physicochemical properties. Local geometry. Docking inferred binding propensities. Best performing strategy. The SCDock seed mix strategy led to the best results, highlighting the importance of combining multiple descriptors effectively. The paper also examined 3 docking energy methods, MAXDO, iAttract, and PISA. Here's how they differ. MAXDO and iAttract, similar in contributions but differ in how they handle clashes and electrostatics. PISA, focuses on the likelihood of a macromolecular assembly being functionally relevant. Understanding these differences is crucial for selecting the right method for specific protein interactions. In conclusion, this research not only enhances our understanding of protein interactions but also provides a framework for discovering new partners and their complex structures. As we continue to explore the vast world of proteins, studies like this pave the way for innovative solutions in medicine and biotechnology. If you found this summary helpful, don't forget to like, subscribe, and hit the notification bell for more insights into the fascinating world of biology. Let us know in the comments what topics you'd like us to cover next. Thanks for watching.
Pharmapal.